Hi, in this chapter, which is chapter 4, we're going to talk about prokaryotic diversity. Habitats, excuse me, and functions of some of your prokaryotic groups. Number one, they're everywhere. So we say they're ubiquitous. They're in the soil, they're in the water, they're up in the air. They oftentimes, it's going to depend on which specific one you're talking about, can survive in extreme cold, extreme heat, extreme pressure. So they're used to growing in a harsh environment. They help to stabilize the ecosystems. They're involved with the nutrient cycling. A lot of them are very adaptable to a changing environment, very resilient. We use them for bioremediation because some of them are able to easily break down very complicated compounds that are hard for other things to break down, and these are capable of doing that. Some of them are pathogens, some of them are responsible for things like food spoilage. So this just shows an example of one type that's involved with helping with nutrient cycling and helping with plants. If you notice, this is a plant root right here, and you have these little nodules. Those are actually concentrations of bacteria. There's some of the nitrogen-fixing bacteria known as rhizobium that is necessary to be on certain plants, like your legumes, like your peas, chickpeas, things like that, have to have this bacteria associated with its roots in order to grow. Sometimes nitrogen in the soil is in a form that the plant cannot use directly. And so what happens is these bacteria, the rhizobium, is able to use that nitrogen that's found in the soil that the plant can't use, and then it converts it into a form that now the plant can use. So they're helpful with the growing of the plants. So there are several cases where we see various types of symbiotic relationships, and we give names for all of them. Mutualism is a type of symbiotic relationship where both the organisms benefit from the situation. Ammennialism, one is harmed, one's unaffected. If one is benefited and the other one is unaffected, it's commensalism. Neutralism, they're living together, but so be it, neither one's being affected. And parasitism is where one's benefited at the expense of the other. The microbiome, we'll talk about that a lot or use that term a lot, and all that's referring to is all the microorganisms that are associated with a particular organism or an environment. So certainly in medical microbiology, we often talk about the microbiome on the human. So we're talking about all the organisms associated with humans in that case. We have a lot of organisms on us and in us that are beneficial. So that's what we're talking about, all these players in this game, if you will. It's the microbiome. The human microbiome, it's going to change over time. Shortly after birth, as the organisms start to colonize on the skin, the intestines, etc., that's going to change over time. Diet can play a role. Stress can play a role. We tend to, with the human microbiome, refer to two subgroups within that, the resident versus the transient microbiota. The resident ones, they're always there, either on the surface or inside the body. Transient ones, as the name implies, they're just there temporarily. Diet may change and then they leave. Stress may occur and then they may show up. So they're not there the whole time. So how do we classify and identify organisms? We're going to look at several different things in the field of taxonomy and systematics. Number one, you're going to look at the morphology. You're going to look at staining patterns. You're going to look at biochemical tests. You're going to look at physiological tests. How does the organism respond under different situations? You're going to also now, as over time as our technology has increased, we certainly do a lot more nucleotide sequencing now and compare like the DNA and the sequence of the bases on the DNA. How similar are the more closely related two organisms are to each other, then the more similar that nucleotide sequence is going to be. So when we classify, remember we're looking at just prokaryotics today. When we classify them, we're going to classify, number one, you've got your gram-positive bacteria. Within that, we have a subgroup. We will divide it by low GC versus high GC. This GC is referring to the guanine cytosine concentration. For the gram-negatives, they're first going to be divided into one of three groups. Either the proteobacteria, what sometimes is referred to as the CFB, the cytophagobacterium and bacteroids, and then the third group are your spirochetes. We have another group of prokaryotes known as the deeply branching bacteria, and then we have our atypical bacteria, which will include your mycoplasma, your chlamydia, and your rickettsias. So rickettsias is just one of the groups. You can see here under the staining of the cell, you can see these smaller purple, those are the rickettsias. They kind of look like little spirals there. They can cause certain diseases like Rocky Mountain spotted fever, where they infect ticks. If we look at the gram-negative bacteria, you have the group, the proteobacteria. These are going to be divided into 5 different classes. You have the alpha proteobacteria, beta proteobacteria, gamma proteobacteria, delta proteobacteria, and then epsilon proteobacteria. So all of these are within the same group of gram-negative. Your alpha proteobacteria, these are ligotrophs. That means they can live in very low nutrient environment. They're not picky eaters, essentially. Some of these are going to be obligate intracellular pathogens, meaning they must grow inside of a host cell. Some examples of these are going to be your rickettsias that we just saw pictures of, chlamydia, agrobacterium, bartonella, brucella, caelobacterium, and rhizobium. In terms of these specific bacteria and what diseases they cause, those will be covered in both Unit 4 and 5 at the end of the semester. The beta proteobacteria, these are eutrophs. That means they require a lot of organic nutrients. These are the picky eaters. Examples are Bordetella, Burkholderia, Leptothrix, Neisseria, and Thiobacillus. Here is Neisseria. It is growing on what is known as a chocolate agar plate. It needs a lot of extra nutrients in order to grow. Your gamma proteobacteria, this is a huge, diverse group. It does include your enterics. Remember, all of these are gram-negatives. Your enterics include your coliform and non-coliform. Enterics are typically those bacteria that they normally reside in the intestines or in the colon. Examples of these would be like Enterobacter, Escherichia, over here, Escherichia coli, that is, we tend to abbreviate it just as E. coli, which probably most of you are familiar with, Klebsiella, Pseudomonas, which is commonly found in the soil, Vibrio, Yersinia. Like I say, it's a very large group. This is showing a picture of one of the members of this group, which is Legionella. It is a rod. Like I said, they're all gram-negatives. This is associated with air conditioning units. So it will live, if you're not familiar with air conditioning units, part of the cooling process uses water. And when this organism was discovered, it was actually during an American Legion convention. A lot of the members got sick. They were trying to find out what it was that was making everyone sick. Your first thought is food at a convention. But they ruled out that it was food poisoning. And so what they finally figured out, that the building where the convention was being held, the air conditioning unit that has this water in it, well, the water wasn't draining very often, and so it was contaminated with this bacteria, which is then blowing out into the room when the blower's on. Since that time when they found it, there are new laws where, certainly in commercial air conditioning units, that water must be completely exchanged at least every 8 hours so that you don't have the water sitting there getting contaminated. Here's salmonella. Salmonella can cause several different things. This particular species, Salmonella typhi, is a causative agent of typhoid fever. It can also cause, as probably most of you have, hopefully not personally familiar with, a type of food poisoning. But it is rod-shaped and once again gram-negative. Your delta proteobacteria is still part of that gram-negative group. This is a small group of bacteria. Some of your examples are Bordetella vibrio, which is actually, it's a small bacteria that actually is a parasite on other bacteria. Desulfur vibrio reduces sulfur. This has been used in bioremediation studies to break down toxic waste, to break down radioactive waste. So it's showing lots of promise as a bioremediation agent. Myxobacterium is commonly found in the soil. Here's a Myxobacterium sample. Epsilon proteobacteria, this is the smallest of those 5 groups. It's microaerophilic, which means it can grow with oxygen, just doesn't like a whole lot of it. Campylobacter is an example. This causes gastroenteritis, which is basically diarrhea, abdominal cramps, things like that. And then Helicobacter is another one. H. pylori, Helicobacter pylori, is known to be associated with stomach ulcers. We'll study that when we talk about the various diseases. This is a picture of it right here. It's also been associated not only with stomach ulcers, but there seems to be a link with stomach cancer. Spirochetes are another group of your gram-negative bacteria. They are motile with what's known as an axial filament, which just means, if the word spirochetes here is a bacteria, axial filament just means it's kind of wrapped in a coil shape around the bacteria. These are really hard to culture. So usually when you are looking for these, you'll usually use dark field because it's going to be easier to see them that way, where the background is stained and then the cells are colorless. Some examples are Borrelia, which is a causative agent of Lyme disease. That number of cases have been increasing over the past several years. And the other, probably more common one, is Treponema, which is a causative agent of syphilis. So like I was saying, it's usually easier to see these under dark field because when the background is dark and the cell is colorless, it's just easier to see them that way. And then your CFB group, Cytophaga, Fusobacterium, and Bacteroides. Cytophaga is aquatic bacteria. Fusobacterium is often found in the mouth, can form a biofilm. So it can be an issue with tooth decay, things like that, gingivitis. Bacteroides is commonly found in your GI tract. So here's an example of Bacteroides. Plactomyces is an aquatic, it's gram-negative. Some of them are swarmers, which meaning they are motile. They will produce what we call a holdfast because they are an aquatic system. They need something to basically hang on to keep them from getting washed downstream. So the holdfast will attach like to a rock or some surface and holds the organism there as the water's passing over. It stays still and, like I said, doesn't get washed downstream. There are some bacteria that are capable of carrying out photosynthesis. These are known as your phototrophic bacteria. They are not algae. They are indeed bacteria. Cyanobacteria is probably one of the most common ones. They're kind of a blue-green bacteria. You also have green sulfur bacteria, green non-sulfur bacteria. As you notice, we're not very creative in the names, because then there's also purple sulfur bacteria and purple non-sulfur bacteria. The sulfur is referring to whether it can use that as an electron acceptor. In this picture, this is showing a container where you've had soil We do this in a general biology class. Sometimes it's known as a Winogradsky column. You put soil in and you get different layering effects. And you can often see the purple and the green bacteria. The purple one is kind of hard to see, but it is a darker purplish color. And the green bacteria gets its name because it actually looks green when it's growing. These are commonly found in the, like on the surface of the soil, sometimes in water. They can carry out photosynthesis. This is showing the cyanobacteria, the blue-green bacteria. These are sometimes responsible for when they grow very rapidly in an aquatic system where you get kind of like this bloom, this massive growth in this pond here. You can see that the surface looks green because of it growing. One thing you do want to just be a little careful of, whether it's cyanobacteria or whether it's algae, oftentimes these will produce toxins. So you don't, other than, I don't know, thinking it's sort of gross going in green water like that, there could be toxins there. And so sometimes it's associated with fish kills, and just be careful around it. With your gram-positive bacteria, this is a large group as well. It is divided into either high or low GC count. And like I said, this is referring to how much of the DNA, what percentage is guanine and cytosine. Remember, we haven't really covered it yet, but we will. There are 4 bases in the DNA strand. You have adenine, thymine, cytosine, and guanine. Cytosine and guanine will always pair together. And so you can do your sequencing, nucleotide sequencing, and determine how much of the DNA is guanine and cytosine, G and C. If it's greater than 50%, it's considered high. If it's less than 50%, it's considered low. So some of your high GC gram-positive bacteria, and I'll be honest with you right now, throughout this course, especially in the lab, you're going to become familiar with the bacteria that we work with, and right now you might be panicking. Don't. There's a lot of names in here. There are some that you'll see more often than others, and just with practice and over time, you'll come to know that, say, E. coli is a gram-negative, Streptococcus is a gram-positive. Am I going to require that you know, if I were to ask you, Corynebacterium right here, is a high or a low GC gram-positive bacteria? No, I'm going to be more concerned about, do you know what disease it can cause? But I'm not going to ask you, is it high or low gram-positive? Just to give you an example of some of your gram-positives here, Actinomyces, Corynebacterium, we'll see that, some Mycobacterium, Nocardia, Streptomyces. And then this is showing a picture. There's Actinomyces, Corynebacterium is over here, and you can see some others in this tissue slide. Some of the low GC ones are things like Bacillus, causative agent of anthrax, Clostridium, causative agent. This has several different things. Clostridium perfringens, which causes gas gangrene. Clostridium tetani, causative agent of tetanus. Clostridium botulinum, causative agent of botulism. And Clostridium difficile, also known as C. diff, which causes colitis. Both Bacillus and Clostridium are commonly found in the soil. And then you have things like Lactobacillus, Staphylococcus, if you've heard of a staph infection, if you've heard of MRSA, Streptococcus, strep throat, things like that. So these are all gram-positives. Here is actually a picture of C. diff, Clostridium difficile. If you work in a nursing home or in the hospital, unfortunately, you're probably going to deal with this. You will know it because it has a distinctive smell. Not so good. Over here under A, this is Streptococcus pyogenes. It's gram-positive. It's usually in a chain like this, coxal shape. And then this is the same organism growing on a blood plate. This petri dish, you literally put red blood cells in it, so normally it looks like this deep red color. When this is growing on it, this particular species, it lyses the red blood cells or clears it. I have seen where you could actually, it clears it enough you can read a paper under the plate. You don't want to see this. It is actually growing here, but the difference in the coloration, it's breaking down the red blood cells. That's not a good sign. If we were in the lab and somebody had a plate where you had clearing like this, we'll just say, I would be sealing that plate up and telling you not to open it. Over here, this plate actually is a mixed plate. We have gram-positive bacillus. That's what the rod, these chains are. It looks like a link of sausages. That's the bacillus. Each little tiny link is an individual cell. And then in the background here, the red is E. coli, which is a gram-negative. Bacillus is gram-positive. On this plate here is the bacillus again. It's growing on a blood plate. Bacillus, oftentimes, from my experience growing it on plates, it usually gets nice, thick, almost like a swarm of spread out, thick growth. This is Staphylococcus. The name coccus tells you that it's spherical or round in shape. Staphylococcus is notorious. One unique thing is that it tends to grow in these clusters. It's described as being grape-like. Some of your deeply branching bacteria, this group is, from an evolutionary standpoint, very important because we feel it's one of the oldest forms of organisms found. They tend to be thermophiles or hypothermophiles, meaning they can grow easily in very high temperatures. And then you have listed here some examples like aquifex of it. And the deep branching ones here. On this phylogenetic tree, the closer you come down here to the base is the first forms of life. So newer ones as you branch out on the edges of the tree, if you will, are newer forms. So you can see this is a common ancestor for the bacteria. Over here, your gram-positives, your gram-negatives are over here. And then you've got your archaeas over here and the eukarya way over here on the right. Here's an example of one of those. The Deinococcus tends to be able to survive under very, very harsh environments. Now we're talking about the prokaryotes. All the other ones we've talked about previously have been under the domain bacteria. Those aren't the only prokaryotes. Remember your archaea. You have three domains, your bacteria, your archaea, and the eukarya. The prokaryotes are in the domains bacteria and the archaea. So don't forget about the archaea. They do exist. We tend to kind of ignore them oftentimes in an introductory course or in a medical microbiology course because none of them are pathogens. But don't forget that they exist. Okay? I mean, that's a good thing. They don't want to kill us. They include methanogens, those that produce methane gas. It's a very wide group. They are single cell. They are prokaryotes. Remember, their cell wall does not have peptidoglycan in it. A lot of these are what we would call extremophiles. They're able to grow in very, very harsh environments. Remember, these are the ones that are considered to be the first living organisms on Earth when the environment was very harsh. They tend to be able to withstand very extreme high temperatures. They can withstand high salt concentrations that would kill most anything else. They can withstand very low pH. So they're, when we say extremophiles, it means they can adapt and survive in a very, very extreme, harsh environment. But the good news is none of them are pathogens. So this just shows an example of some referred to as halobacteria. They are able to grow in, these are salt ponds, so it's where the water and the pollen have very high salt concentration. They are able to survive in there. Most things could not survive that salt concentration. It would kill them, but the archaeas are able to survive there. So this is just a breakdown of your prokaryotic cells.